Hi guys, we are starting Unit 3 today with Chapter 10, um, the sensory function of the nervous system. So we're going to go over, actually today's lecture is separate into two parts, Part 1 and Part 2 within the same recording. So Part 1, we're going to go over sensory physiology, understanding how information is being transmitted to the central nervous system from the peripheral nervous system. So there are sensors, there are receptors in the periphery who are capable of detecting specific uh, sensory signals, different types of sensory modalities, and then send it to the central nervous system for us to perceive our reality. We're going to go over, before we begin the lecture, we're going to define some two additional graded potential. So far we've learned from graded potentials are EPSPs and IPSPs. Today we're going to add two more, generator potentials and receptor potentials. And you've seen these receptors from Bio-31 anatomy. For example, the Pacinian corpuscles, also known as lamellated corpuscles. These Pacinian corpuscles, they detect mechanical stimuli. For example, deep pressure on the surface of your skin. So as your skin receives a deep pressure stimulus, they can distort skin areas deep enough all the way down to the Pacinian corpuscle. And once the sensory part of the receptor is distorted, they're gonna induce the axon here, part of the first order neuron called afferent neuron, or first order neuron or ascending neuron, which is a sensory neuron, to fire action potentials and propagate that action potential toward the spinal cord, right? So this sensory neuron, this first order neuron, has its cell body located in the dorsal root ganglion, right? So one ganglion, two or more ganglia, dorsal root ganglion or posterior root ganglion. That ganglion, each ganglion contains hundreds of cell bodies of first order neurons, one of which has its peripheral axon all the way and wrapped by a structure that we call the Pacinian corpuscle. And when you excite the Pacinian corpuscle induces the first sensory neuron to fire an action potential. So at the distal part of this sensory neuron, there's a trigger zone, and it's the same thing. In order for that trigger zone to fire an action potential, the threshold potential needs to be met, right? So the Pacinian corpuscle here will basically generate generated potential, and they add up. All these generated potential being graded potential, they add up, they help each other, and all of them, all the summation being added to one another, and if they rises above the threshold, the trigger zone of the first order neuron will fire that action potential, and that is called generated potential. Another type of graded potential we call receptor potential down here. So this here, for the Pacinian corpuscle, this, for example, for deep pressure, so this would fall under one of the four general sense. Right. So one of the four general sense being pressure, and then Pacini corpuscle are basically conveying pressure perception to the brain. The next one, the receptor potential, this is used by special senses. There are 5 special senses. There are 4 general senses. You should have learned that from anatomy. And there are 5 general senses. For example, here, this is showing you a sense of taste, right? So when you're tasting food, this what goes on in your taste buds, it's called the receptor potential. The reason is the neuron, the sensory neuron, there's a space, microscopic space between the sensory neuron and the peripheral receptor. In this case, a taste receptor. So the stimulus is a chemical from food that has been dissolved in water. If the food chemical is not dissolved in water from your saliva, you cannot taste that chemical. So tasting requires chemicals to be in solution with the water from your saliva. So when it's dissolved in that water, it can be recognized by a sensory receptor, specifically a taste receptor. So the stimulus here binds to the receptor, and you know the story. It leads to depolarization of the cell membrane, the plasma membrane, the phospholipid bilayer of this sensory receptor. And that depolarization will activate a voltage sensitive calcium channel. And that voltage sensitive calcium channel opens, leading to a massive influx of calcium, divalent cations, and the influx of calcium induces vesicles containing the neurotransmitters to fuse with the cell membrane, plasma membrane, and empty their contents into that microscopic space. So the neurotransmitter diffuses across the space by simple diffusion, driven by concentration gradient, binds to its receptor on the first order neuron, right, on the sensory neuron. And this receptor is a ligand gated, a neurotransmitter gated ion channels. And it will generate a graded potential. We call it, the receptor potential is actually generated by the sensory receptor. And the receptor protein here, this is a graded potential. This is the regular, very similar to EPSPs and IPSPs. So if it's EPSPs, it will need to go above threshold before the trigger zone will fire that action potential being transmitted toward the central nervous system, right? So this is a mechanism by which we taste our food. And talking about taste, when you taste your food, 80% of your ability to taste your food is dictated by your sense of smell. So if your sense of smell is being compromised, most of your ability to taste your food will also be gone, right? And we're going to go back to that later when we get to the rest of the lecture. So that's the 2 additional graded potential, generated potential, and receptor potential. And then we're going to go over, understand how receptor physiology functions. Most of it is basically, we covered the foundation already. We're just applying here. And then introducing a brand new concept, label lines. This also should have been covered in anatomy. well, at least my anatomy classes, I introduce my students to this. So sensory pathways involving label lines and the concept of sensory units versus receptive fields. And then very, very beautiful concept, sensory coding. Um, how does the brain understand the nuances of these variety of modalities, even though we have only one single prototype of action potential, right? One intensity. How is that information being sent and decoded by the brain for perceiving our sensory pathways, like the four general senses, for conscious awareness of the four general senses and conscious awareness of the five special senses. All of that requires proper sensory coding. So that's part of today's lecture. So with that said, let's get this thing started. Need the space where. Okay, let's start with this first. Receptor adaptation. There are certain peripheral receptors that are capable of remaining active. This is the green tracing. The receptor, it remains active as long as the stimulus is present, which is the pink line down here. So for the pink line, the stimulus is on, so it's activating the receptor. And here, stimulus off means the stimulus goes away. So when it's on, you see this pink line, a positive deflection. And this is the receptor responding through a potential, right? So you see that potential above threshold. This is the baseline here. That's the threshold. So the potential is above threshold throughout the same duration as the presence of stimulus. We call those responses, those receptors, slowly adapting receptor. And when we say slowly adapting receptor, they are also known as tonic receptor. They remain active as long as the stimulus is present, as long as the sensory modality is present. So specific examples would be muscle stretch receptors. We're going to revisit them when we talk about the muscle unit, and Merkel discs in our cutaneous membrane. So those two are basically relying on slowly adapting receptors to detect the presence of a stimulus, to detect the adequate stimulus, to detect the proper sensory modality. So for example, the muscle stretch receptor detects the length of the muscle to protect muscle tear, and the Merkel disc detect pressure, for example, touch. So those are examples of slowly adapting responses or sensory receptors. There's another type of receptor adaptation, and that's over here. The receptor only responds to change, on and off, right? That's what we call rapidly adapting or phasic receptor. they respond only when there's a change in on and off signal. So when the stimulus comes on, they detect it. When the stimulus comes off, they also detect it. For the remaining period where the stimulus is present, they don't respond. You see the flat line right there, right? So they respond only at the beginning of the stimulus and only at the end of the stimulus. So when the stimulus is on and when the stimulus is off, they respond. But throughout the rest of the duration for that stimulus, it's a baseline, so they ignore it. And we call that rapidly adapting receptors. And examples of receptors working with this mechanism: receptor detecting smell. So we have olfactory receptors, or receptors detecting deep pressure, Pacinian corpuscle. So those are distinct receptor responses. Either some type they remain active as long as the stimulus is present, and other type they respond only to changes, meaning the appearance of the stimulus at the beginning and the end of the stimulus when the stimulus is no longer present. The concept of a labeled lines, even though we talk about this, it should have been covered in anatomy, but not in this context. But the structural neurons should have been covered. This is called a sensory pathway, and it involves 3 neurons, sensory neurons in series, meaning one after another, starting with the first order neuron. So the first order neuron is in blue, which is a sensory neuron belonging to the peripheral nervous system, because this cell body here, they are aggregated in the DRG, the dorsal root ganglion, right? So each dorsal root ganglion consists of hundreds of cell bodies of these first order neurons. And these first order neurons are classified as part of the peripheral nervous system. So these first order neurons are peripheral neurons. They detect the stimuli, their respective sensory modality. They generate graded potentials arriving at the trigger zone. And if it's above threshold, the trigger zone will fire an action potential and transmit it toward the spinal cord. Once they're inside the spinal cord, they will synapse with another sensory neuron. And the second sensory neuron is shown in green. And this is called a second order neuron. And this synapse is in the spinal cord, right? The synapse here, spinal cord or the brainstem. And it's not random, it's not anywhere in the spinal cord. It is within the sensory nuclei. So this would be how do I say this? Posterior gray horn of the spinal cord, or dorsal gray horn of the spinal cord. Right? But I prefer posterior because we are walking on only 2 limbs, A four-limb animal would be applicable for ventral and dorsal. In case of humans, it's anterior and posterior. So let's call that posterior, a gray horn of the spinal cord. And more specifically, is the distal posterior gray horn of the spinal cord. Because there's distal posterior gray horn and proximal posterior gray horn. Okay, and the second order neuron will convey an action potential toward the thalami. So there's the thalamus here. There's actually two of them, right? There should be a right and left thalami. And this is where you have the third order neuron. So you have the thalamus, right and left thalamus, and you have the third order neuron connecting each thalamus to the cerebral cortex. When we say cortex, it's specifically, we're talking about, in this case, the cerebral cortex, and more specifically, it's the primary somatosensory cortex, okay? Primary somatosensory cortex. That's the functional name. The anatomical name is the post-central gyrus. So it's the same area of the brain, same area of our cerebral cortex, having two names, anatomical / structural name called post-central gyrus, and functional name, primary sensory cortex. And this is where you perceive your reality. This is where a sensation becomes a perception. This is where a sensory stimulus becomes a perception. This is where you become consciously aware of a sensory stimulus. So if the information, if the signal, if the sensation fails to get to the primary somatosensory cortex, the individual is oblivious to the existence of that stimulus. So from the peripheral receptor, right, all the way to the thalami, that stimulus, it's called a sensation. And when that sensation is being transmitted by the third order neuron and go to the primary somatosensory cortex and received by these cortical neurons, we have perception. And so this is when sensation is being promoted to a perception status. So if any information, any sensory information failing to go to the cerebral cortex, in this case, specifically the primary somatosensory cortex, then there's no perception involved. So that's why we call it 3 neuron pathways: first order neuron, second order neuron, and the third order neuron. Among the 3 neurons, only the first order neuron is a peripheral neuron. The second order neuron and the third order neuron, they are both central neurons. The second order neuron and the third order neuron are classified as part of the central nervous system. Moving on to what is called a sensory unit. This is being defined as one sensory, one single first order neuron. So one afferent, one first order neuron, one sensory neuron, and all the receptor that is it's innervating or it's serving, all the receptors are sending information to it, right? We call it a sensory unit. So units with generated potential, units with receptor potential. So this generated potential is this one, and receptor potential is this one. So that's a sensory unit. So you have one neuron innervating a surface in the peripheral nervous system. And we call it sensory unit. And down here, the concept of a receptive field. Receptive field is used to by three specific modalities. It is used by tactile, meaning sense of touch. Is used by proprioception, meaning receptions in your joints, for example. It allows you to know your body positioning in space in your surrounding. So tactile, proprioception, and also vision, meaning sense of sight. So those are the three specific sensory pathways relying on what is called a receptive field. And in the case of vision, we don't call it receptive field, we call it visual fields. But the concept is the same. It allows you to locate the stimulus with highly accurate localization capability. So let's say you have a paper cut, you know exactly where the paper cut is on the surface of your body, right? When you walk, for example, and then you know, let's say you have your lower limbs, one is on the bed, the other one is on the floor, because of the proprioception, it allows you to perceive, for example, if your right lower limb is on the floor, because of the joints and the pressure by the joint due to the harder floor, you know that you're on the floor, and you know the consequence of it. If you jump on it, it's going to hurt. Whereas on the mattress, it would hurt less because there's no such pressure input, putting us much less stressful on your joints, for example, your knee joint, from the mattress than from the floor. That's due to proprioception. So that's the concept of receptive field. Now, what is important for the receptive field is, this is the entire surface area of the skin, for example, right? So if you have a stimulus here and a stimulus here and a stimulus here, right? So even though the three stimulus come from three different areas on the surface of your skin, your brain will not be able to distinguish them as three different stimuli coming from three different areas in your skin. As far as your brain is concerned, in this case, it comes as the same stimulus. In other words, if you touch your friend, in this case here, three different locations, your friend will say it's the same location. So that's the caveat of this receptive field concept. So the larger the receptive field, the capability of the brain to distinguish two stimulus coming from two distinct areas would be compromised. So in order for the brain to distinguish highly accurate two stimuli as distinct stimuli, you need to have a small tiny receptive field. We're going to come back to that concept very shortly. Okay. Let's go over sensory coding. And this is important. So when we talk about action potential, all action potential are the same. They have the same intensity. There's nothing interesting about them. They're really, really boring in terms of variety. There's only one variety. There's not much diversity. It's just one action potential. So because of this, how does the brain decode all of this? Okay, and that's an important thing, and we call that sensory coding. So we're going to go over this concept, sensory coding. First of all, how does the type of stimulus is being coded? Right, so this is depending on the receptor. So modality-dependent sensory receptor allows the brain to perceive different types of stimulus. Whether it's the pressure, so it's detected by the Pacinian corpuscle. Whether it's a chemical from food, then it's coming from a taste receptor in your mouth. So modality-dependent sensory receptor. And modality here means energy, a form of stimulus, or the type of stimulus. Now, it takes two to tango, right? It requires the stimulus, the presence of the stimulus, but also it requires the specific receptor that is tuned to that stimulus. So for example, here, this stimulus needs to be recognized by this particular receptor cell. So the chemical from food would not be detected but by the Pacinian corpuscle and vice versa. The mechanical vibration from detected by the Pacinian corpuscle would not be detected by the taste receptor on your tongue. So not only you need the presence of stimulus, but you need to have a compatible sensor, a compatible receptor that is capable of recognizing and be activating by that modality. Okay. That's why we have the 9 senses. We have 4 general sense and 5 special senses, a total of 9 senses that we can detect to perceive our reality. If there's a modality, meaning a stimulus that is there and there's no corresponding receptor, we are completely oblivious about the existence of that stimulus, of that modality. So we call that so this is how the brain recognizes the type of the stimulus. Due to the existence of different types of receptors, different types of sensory receptors capable of recognizing a very specific type of modality. Now, in addition to the type, how does the brain decode, for example, the intensity of a stimulus? So coding for stimulus intensity can be done in 2 ways. Frequency coding. This word here. For stimulus intensity. But also another way, stimulus intensity also down here, by population coding. So both frequency coding and population coding allows the brain to determine the intensity of a stimulus. So let's start with the stimulus intensity, the frequency coding, meaning the number of action potential being fired, being generated per unit time. So you see here, two stimuli. There's one here, and there's another one over here. The green is stronger, right? So it has, so stimulus strength, this is stronger. This is a stronger stimulus. This is a weaker stimulus, right? W for weak, S for strong, right? So the weaker stimulus will generate a smaller graded potential. So the receptor potential here this is a GP. So it's a graded potential. Specifically receptor potential, right? So this is would be a small graded potential. But the stronger stimulus would generate, this is also a graded potential, a larger L graded potential. And what's going to happen is the trigger zone will generate here 1, 2, 3, 4, 5, 6 action potential. 6 APs. Over here we have 1, 2, 3, 4, 5, 6, 7, 8, 9 APs. So as you can see, the number of action potential per unit time is proportional to the strength of the stimulus, right? Stronger signal, stronger stimulus, larger graded potential, higher number of action potential generated by unit time. So let's say here, so we are clear, this, for example, is within 5 seconds. Same thing here. This would be also within the same 5 seconds, right? So 6 action potential per 5 seconds or per second, and 9 action potential per 5 seconds or per second. So it has to be the same duration, yet the number of action potential being generated has increased due to the stronger stimulus, producing a larger graded potential, leading to an increased number of action potential generated by the trigger zone and propagated down these node Ranvier. And so that we learned already. So what is new is the coding, right? Based on the frequency of firing of action potentials. Frequency means here, again, the number of action potentials being generated per unit time. And that per unit time, you can express it per second, you can express it per minute, you can express it per hour or per day. It's up to you, you decide. But if you are comparing, then you have to express the same unit, right? So over here, if you say per second, then over here, you have to be also per second. If you don't, then you have to convert it per second. So when you compare, you have to compare apples to apples. So you need to compare time unit that are similar. You cannot compare per second with per minute, obviously, right? So that's what I mean by that. So frequency coding is the exact number of action potentials being produced at the trigger zone per unit time. And we call it frequency coding. And that contains information about the strength of a stimulus. And we call it stimulus intensity. So that's for frequency coding. We also have another way for the brain to recognize the strength of a stimulus. And down here is population coding. So for example, this is a single sensory unit, meaning one sensory neuron serving this particular area of the skin. So a weak stimulus would activate only 1, 2, 3, 4 peripheral endings, right? So these can be called bare nerve endings or free nerve endings. So you or axon terminals, if you will. So these are only 4 with a weak stimulus. A stronger stimulus would mobilize a greater number of endings. So here's only 4, but here's all available endings from that sensory neurons would be activated, right? Population coding. Here we have multiple sensory units, specifically 3 different first order neurons serving alongside each other on a specific defined area on the surface of the skin. A weak stimulus might mobilize only the middle neuron, but a strong stimulus mobilizes all 3 sensory neurons. So the strength of the stimulus would be directly proportional to the number of graded potential being generated. The greater the graded potential being generated, the greater the chance of triggering an axon potential generated at the trigger zone and send that to the central nervous system. So the stronger the stimulus, the greater the chance of generating action potential and being propagated down toward the spinal cord or the central nervous system. Okay. So that's population coding versus frequency coding. Another one is for allowing the brain to locate the exact position of the stimulus, meaning where does the stimulus comes from. For example, you have a paper cut, you know exactly where that paper cut is. So one way is the same 3 neurons from the previous slide. This here, these 3 neurons, or these 3 neurons, right? And they're being magnified. And this is how the brain can basically fine tune the signal. So, for example, the stimulus is activating specifically the pink neuron, the middle neuron, Y1, right? It's activating Y1. And so the brain, in order for the nervous system to locate with accuracy, what's going to happen is once the action potential travels down its axon and going toward Y2, which is the second order neuron. So Y1 is the first order neuron, Y2 is the second order neuron. And that Y1 sprouts out collaterals, synapsing with interneurons on either side. And these inhibitory interneurons will basically induce Z1 to release less neurotransmitters synapsing with Z2. Same thing here, inducing X1 to release less neurotransmitters into the synapse with X2. So here, transmission has been decreased significantly, whereas the action potential on this one is amplified, allowed to go to the brain. So the brain perceives the stimulus coming from this area. So this area has been attenuated. This area has been attenuated by the concept called lateral inhibition. This is an amazing feature because here, that's a potential by sensory neurons, the first order neurons, X1, Y1, Z1. But here is the same thing, but by second order neuron, action potential being generated, Y2 significantly more than X2 and Z2, because X2 and Z2 have been prevented significantly from generating action potentials due to these inhibitory interneurons. And you should be able to answer that question by the specific mechanism, and we all learned that. How is it possible for Y1 to induce the interneuron here to go on to induce this axon terminal to release less neurotransmitter, right? So the interaction between the inhibitory interneuron and Z1, we learned that concept, right? So that's basically presynaptic inhibition. Same thing here. This inhibitory interneuron with X1, that's presynaptic inhibition, decreasing X1 from releasing it, or at least, yes, inhibiting X1 from releasing its neurotransmitter, inhibiting Z1 from releasing its neurotransmitter. So there's less neurotransmitter from X1 to activate X2, less neurotransmitter from Z1 activating Z2, right? Less neurotransmitter, less graded potential. Less neurotransmitter, less graded potential. Less graded potential, the chance of fine action potential decreases by the axon hillock of X2. Same thing here. Axon hillock of Z2 also will fail to find action potential due to a less weaker signal coming from the graded potential due to a decrease in number of neurotransmitters being released by Z1 and X1 respectively. So by using this lateral inhibition mechanism, the brain can locate with accuracy where is the signal, especially if for example, if it's a cut, right? If it's a traumatic tissue injury, the brain needs to know the exact location, and lateral inhibition is one such mechanism used to allow the brain to fine-tune, to locate exactly where that noxious stimulus comes from. And on a very similarly related concept called tactile acuity, two-point discrimination. So let's say you have two stimulus, one right here and one right here. This large receptive field, the brain will perceive it as one same stimulus. But over here, since two different neurons, you have a stimulus here and stimulus here, the brain will be able to perceive as two different positions on the surface, the same size area on the surface of the skin. So this one have a higher acuity or acuity. I don't know how to pronounce this word. Okay. So this is higher. So it can have a higher discrimination capability perceiving as 2 points. But with a larger receptive field, we have a problem. Larger, we will have a lower capability. We are compromised in terms of our ability to detect two different stimuli. We will detect that as one. Okay. So with a larger receptive field, our ability to detect two different stimulus decreases significantly, but with a smaller receptive field, our capability of distinguishing 2 points are highly accurate. So this changes dramatic over the surface of our body. For example, our lips, they have a very small receptive field. Same with the tip of our fingers. But let's say on our back, we have large receptive field, so we would not be able to detect with much accuracy on the back, on our back, for example. Okay. So that's the concept of 2-point discrimination. For 2-point discrimination, you want small receptive field. The smaller the receptive field, the better your capability of detecting 2-point discrimination. Moving on to the second part of the recording. So briefly, organs of general sense. So pain, temperature, pressure, and touch, change chemicals here to touch. Okay. So pain, temperature, pressure, touch. They have common features. Their receptor has a simple structural design. So their structure is really simple. They also have a widespread distribution. So the receptors are all over, for example, the surface of your skin, right? Pacinian corpuscle is in your dermis. Meissner's corpuscle is at the boundary between the dermis and the epidermis. The free nerve endings are throughout the epidermis. All of them have very simple structures, but they have a widespread distribution. That's organ for the 4 general sense. So we have 4 general sense. Sense of pain, temperature, pressure, and touch. We have 4 general sense, and we have 5 special sense. These special sense, their structure is really complex, and they have a restricted distribution. You don't have 1,000 eyes, right? You have only 2. And the eyeballs is a very complicated organ compared to the Pacinian corpuscle, right? So as far as the general sense concerned, complex structural design and restricted receptor distribution, okay? So we're going to go over, the special senses. So for example, and sense of smell, you recall to know that's olfaction. Sense of taste, gustation. Sense of hearing, sense of balance, equilibrium. So these are the nerves. So the stimulus, for example, sense of smell, the stimulus is chemicals. The receptor is called olfactory epithelium, sending information through cranial nerve number 1, olfactory nerve. Gustation, sense of taste. Stimulus, dissolved chemicals from food, like I mentioned earlier. Receptor, taste buds. That information is being sent through cranial nerves number 7, 9, and 10. So sense of taste is conveyed by 3 different cranial nerves, 3 different pairs of cranial nerves, right? Pair number 7, pair number 9, pair number 10. Sound of the sense of hearing. Stimulus is sound waves. Receptor is cochlea, sent through nerve number 8. And equilibrium, sense of balance. Body positioning, receptor vestibular complex, cranial nerve number 8 as well. What is unique about the sense of hearing and sense of balance? The stimulus are mechanical. So they are activated by mechanical stimuli. Whereas olfaction and smell the stimulus here for sense of smell and sense of taste, the stimulus is chemical. So they would deal with chemical stimuli. So mechanical stimuli versus chemical stimuli. Whereas vision, again, it's a little bit different. This is called radiant. radiant, radiant energy, or visible light, right? So coming from sunlight. So this is radiant energy, this is chemical energy, this is mechanical energy. So different stimuli, different modalities. Both the sense of smell and sense of taste, the chemical needs to be dissolved. If you are the olfactory epithelium is completely dry, you will not be able to smell anything, right? So if you have your olfactory epithelium completely burned because you used to sniff, for example, highly toxic acidic chemicals like hydrochloric acid, then it burns off completely your olfactory epithelium, then you lose the ability to smell for the rest of your life. And we mentioned earlier, your ability to smell dictates 80% of your ability to taste. So when you are sick, that olfactory epithelium is inflamed, right? It secretes excessive mucus, compromising your ability to detect chemicals, so compromising your sense of smell. And when you're sick, the sense of smell is compromised, it compromises your ability to taste. That's why when your mom cooks you something when you're sick, your favorite food, even if it's your favorite food, no longer tastes the same. It's because of this fact. So when you have a compromised sense of smell, it will compromise your ability to taste. And among the 5 special senses, the oldest one is the sense of smell. And the sense of smell is the only one bypassing both thalami. Right and left thalami. So 2 thalami. 1 thalamus, but 2 thalami. So that signal, olfactory signal, goes straight to the olfactory sensory cortex. It bypasses the thalami. Because of this, sense of smell is the only sense that has a direct memory component to it. So let's say you go to a foreign country. Let's go, your mother is she's Italian, right? She makes, delicious Italian food. You visit Italy, you walk downtown or, next to a restaurant, and it happens to cook the same meal as your mom used to make. And the same smell, when you smell that, it brings back vivid memory of your childhood with your mother cooking that meal, all that. So only the sense of smell can convey that perception, since it is bypassing the thalami. All other senses, we don't have that direct connection to a memory bank, right? So sense of smell has a direct connection to our past. So it brings back vivid memory when our sense of smell is being activated. So that's what sense of smell being the oldest. The most important sense, however, due to the sheer real estate, is vision. Our sense of sight, 40% of our cerebral cortex is dedicated to digest, to integrate incoming visual stimuli. Almost half of the part that is the most intelligent part of our brain, the cerebral cortex, 40% of it is dedicated to integrate, to deal with visual information. So with that said, vision is the single most important among the 5 senses. There's one more thing about the sense. The general sense, when we talk about this general sense up here, sense of pain is also unique. Perception of pain overrides all other sensory modalities because pain perception protects you from further injuring yourself. This is a life-saving ability. I'll give you an example. Pressure, right? Like a caress. Someone touches you lightly, you might enjoy it. If that same person starts to punch you, then it's less enjoyable. But when that punch becomes a bruise, when there's tissue injury, then you no longer feel the pressure and you no longer feel being touched. You feel the pain. Same thing with temperature. You take a bath, the water initially is cold, it's uncomfortable, right? And then you increase the temperature where it's room temperature, then it becomes comfortable. And when it gets to a certain temperature, you feel it's hot, but past a certain point, let's say exactly at 54 degrees Celsius, you don't see the water as hot anymore. You feel pain. And that point, you lose the ability to appreciate temperature. All you can perceive is pain. In that sense, pain perception overrides all other sensory modality. So let's use 2 pathways. We're going to use pain perception and vision. So pain perception, that's for the general sense. And vision, that's for the special sense, because we don't have time to go over 9 senses. So we're going to go over the most important one, overriding all other senses. So sense of pain, and then vision. Yeah. But before we talk to that, this is basic review here from anatomy. So the receptor that detects noxious stimuli, noxious, N-O-X-I-O-U-S, noxious stimuli will allow you to perceive pain. And the receptor detecting noxious stimuli, they are called nociceptors, okay, leading to pain perception. So temperature extreme, mechanical damage, excessive chemicals that can burn your skin, that will activate the nociceptors. And they're located in skin surface, epidermis, joints, bones, walls of blood vessels. Thermoreceptor detect temperature. So free nerve endings or bare nerve endings in the epidermis, located epidermis rather than dermis, skeletal muscle, liver, and hypothalamus, because hypothalamus is our natural thermostat. So it can reset your body temperature. That's a very important neural structure. Remember this hypothalamus, it also controls the endocrine system, but it is part of the nervous system. Mechanoreceptors, detecting stretch and compression. So tactile receptor like touch, pressure, and vibration. Baroreceptor and pressure. We're going to come back to them when we talk about the cardiovascular system. And then proprioceptors in joints and SKMs in the skeletal muscles. And chemoreceptors, detecting proton concentration, partial pressure of CO2 in the cerebral spinal fluid. This is for the brain to detect the level of CO2. And we're going to learn that too. Surprisingly, the central nervous system is much more sensitive to CO2 than oxygen, because CO2 is directly linked to proton concentration, pH, and pH fluctuation is deadly in the body, so it's maintained vigorously. And because of the relationship between oxygen, carbon dioxide, and pH, that's why the brain is extremely, excessively sensitive to CO2, much more than O2. And also partial pressure of oxygen and partial pressure of CO2 in our blood, arterial vascular bed. So that's, you learn as is, there's nothing cerebral there to explain. Moving on to here, the single most important pathway is the spinal thalamic tract. Let's break down this term, spinal thalamic tract. Spinal mean the pathways begins in the spinal cord. Thalamic, the pathway ends at the thalamus. Right? If the pathway begins in the spinal cord, it ends in the thalamus in the brain, then you know that's a sensory pathway, right? And the word tract, it's an aggregation, bundles of axons that are physically located in the central nervous system. Either these axons run through the spinal cord or the brain, we call it a tract. Bundles of axons running outside of the central nervous system, we call it a nerve, okay? So spinal thalamic tract, you know that is part of the central nervous system. And it is the single most important pathway conveying somatosensory perception. And this pathway goes to the primary somatosensory cortex, the post-central gyrus, right here, primary somatosensory cortex. This is where conscious awareness of a sensation begins. This is where perception starts. That's a very important word, perception. If information fails to get there, there's no perception. Perception, perception. Perception means conscious awareness of a sensation. Now, due to the concept of decausation here, the left primary sensory cortex receives information from the right side of the body and vice versa, right? Because of the fact that sensory information crosses over when it gets into the central nervous system, either at the entry point or like here at the entry point, or it goes up to the brainstem and it crosses over the medulla. Yeah? But either case, it's decausation, so it crosses over. So you're going to learn as is here, proprioception and mechanoreceptors are sent through the dorsal column medial lemniscal pathway, whereas nociceptors and thermoreceptors are sent through the spinothalamic tract. So temperature and pain through the more important pathway, spinothalamic tract. The difference between the two, the spinothalamic tract crosses over at the entry point, whereas the dorsal column pathway moves on the same side all the way up to the medulla, then crosses over to the other side. And we defined modality already early in the lecture, just the different types of stimulus, different types of energy that is used to activate a sensory receptor. And when we deal with pain, there's a somatic pain and visceral or referred pain, mean the same thing. So they are synonymous. You can say visceral or you can say referred pain, means the same thing. And this comes from our visceral organs, right? So that's why it's called visceral here, meaning organs in your thoracic cage, organs in your abdominal cavity, or organs in your pelvic cavity, right? So those are the term visceral organs. Visceral organs in your thoracic cage, in your abdominal cavity, in your pelvic cavity. So thoracic cage, abdominal, pelvic cavity. Any organs within these three cavities, if they are compromised, they will send sensory information. Unfortunately, so here, this is an example. The first order neuron is unique to the heart, sends it to, but that heart, information coming from the heart, does not have an exclusive second order neuron. It shares the second order neuron with, this second order neuron belongs actually to the skin. And that skin happens to be on the area of your left pinky, right? So when the heart is compromised, it's going to activate the second order neuron. But that second order neuron, as far as the brain is concerned, it perceives information coming from your left upper limb, specifically your forearm and your pinky finger. So when the heart is compromised, you have a numbness on your left upper limb because of that, because the two first order neuron shares the same secondary order neuron. And that second order neuron actually belongs to the skin and not to the heart. So that's why we fail to locate with accuracy where the discomfort comes from when any visceral organs has a problem. So if you have a discomfort in your abdominal area, you would be hard pressed to tell your physician exactly where the information comes from. Where's the discomfort? Let's say you can't tell the physician, hey, I have a pain, I know it's from the liver. I have a pain, I know it's from the pancreas. It's not possible because we don't have that direct line of communication from the visceral organ to that primary somatosensory cortex. And we call that visceral pain or referred pain. Whereas somatic pain, every single peripheral location has a direct line to the primary somatosensory cortex. So these are the area of discomfort if you have a problem with these visceral organs. This is a very important concept, the ability to modulate pain. The first one is called gate control theory. This is the C fiber. So this is a specific type of first order neuron, right? So let's say you bump your elbow into a corner. So it's going to trigger action potential sense here, shoots to the brain. So this is the second order neuron, first order neuron, second order neuron. Action potential travels all the way to the primary somatosensory cortex to perceive the pain. And simultaneously, this axon collateral will inhibit this interneuron from firing. So this inhibitory interneuron will fail to release the neurotransmitter here. So it's in the spinal cord. So this neurotransmitter here would be glycine. So the glycine would be prevented from being released. So this cell body with second order neuron just fires action potentials easier. There's no IPSPs, there's only EPSPs, right? So over here is EPSPs, fires action potential. Here, IPSPs by glycine is not there because glycine is not released. But if you start to rub on your elbow, which is here, the pain is modulated. So the elbow has also another type of sensory called AB fiber. It has a larger diameter of axons and is myelinated. So it's going to propagate action potential faster. So the signal arrives before the C fiber. So what's going to happen here is it's going to activate this interneuron. This interneuron fires EPSPs, action potential fires here, glycine release. And if glycine is released, glycine induces IPSPs on the cell body with second order neuron. And when this signal comes in, that cell body has been hyperpolarized. So significantly less action potential be sent toward the central nervous system, leading to perceiving less pain. So this is why we rub our skin area when we bump our elbow into something. The more you rub, the more you lessen the pain by rubbing it because of this gate control theory, right? So the AB fiber activates the inhibitory interneuron, allowing them to release glycine, and the glycine induces the cell body of the second order neuron to generate IPSPs. So the signal coming from the C fiber producing EPSPs would be offset by the IPSPs, so less action potential being fired by the second order neuron. Less signal goes to the brain, less pain perception. Over here, there's no modulation. So the C fiber sends action potential here, EPSPs, and simultaneously inhibits the inhibitory neuron here. So this interneuron will not be firing action potential, no glycine release, no glycine release, no IPSP on the cell body second order neuron. Only EPSPs arrives, most likely action potential will be fired. That's why we perceive the pain without rubbing. And with rubbing, less pain perception. So that's the gate control theory. There's another one. This is inside the spinal cord. This is also more powerful. It's called the endogenous analgesia system. This is ability of the brain to block the pain. So the brain sends signals down to the brainstem, and ultimately at the spinal cord level here, that's where I want you to focus on. The descending neuron will release neurotransmitter activating the interneuron, right? And what is important here is, even though it's not shown, the neurotransmitter here, the descending neuron, will basically induce interneuron and find action potential, inducing release of a novel neurotransmitter called the enkephalin. The enkephalin is a natural opioid, like morphine, but morphine is man-made. Morphine is synthetic, but enkephalin is naturally made neurotransmitter. And that enkephalin has a dual function, two functions. First, it reduces the amount of neurotransmitter released by the afferent neuron here. So this is a sensory neuron. This is the first order neuron, right? So the first order neuron releases substance P, a neurotransmitter. So if the enkephalin is released, it will basically hyperpolarize the axon terminal of the first order neuron, making it releasing less substance P. If you have less substance P, you will have less EPSP generated by the second order neuron. That's one mechanism. The other mechanism, enkephalin is also producing IPSPs on the cell body of the second order neuron. So again, decreasing the probability of firing action potential. Therefore, decreasing the signal going toward the thalami and ultimately to the primary somatosensory cortex, decreasing pain perception, right? So this is a second order neuron here being prevented from firing action potential due to these two mechanisms. So enkephalin released by the interneuron has two functions. Function number one, it inhibits the first order neuron from releasing substance P. And function number two, it induces generations of IPSPs on the cell body of a second order neuron. Right? In both cases, less substance P, less formation of EPSPs on the cell body of the second order neuron, less action potential being fired. The second mechanism, enkephalin induces the formation of IPSP directly on the cell body of the second order neuron, less action potential being formed, less signal being transmitted to the thalami, ultimately less to the primary somatosensory cortices, less pain perception. And that's the second mechanism. So there's two ways to modulate pain. One mechanism is from the periphery, rubbing the elbow. The other one is from the spinal cord, the central mechanism. So one peripheral mechanism, one central mechanism. Now moving on to vision. Panel 1 and panel 3 are review. So this is basically your, you're gonna learn on your own because this is completely anatomy here. So these are the cells in your retina. So light comes this way, goes all the way in, activating the photoreceptors, cone for color vision, rod for black and white, night vision, and information goes back out. So light is radiant energy being converted by the photoreceptors into graded potentials, and the graded potential travels back out onto the bipolar cells and onto the ganglion cells. And the ganglion cells are the only cells, only type of neuron from the retina capable of generating action potentials. So the ganglion cells generate action potential and they send that action potential, they propagate action potential along the optic nerve, cranial nerve number 2, right? So the optic nerve are aggregations, bundles of axons that are located physically outside the central nervous system. That's what we call it, nerve, optic nerve. Okay? And those axons belong to the ganglion cells, the only type of neurons in the retina capable of generating action potentials. Everybody else, every other type in the retina are only producing graded potentials. And this is the neural pathway. So as you go here, you have parallel processing, depth perception, and then, you know, information is at the thalami over here, lateral geniculate of the thalamus, and it will send information to the primary visual cortex located in the occipital lobe. Now, when it gets to the thalamus, information goes actually three pathways: to the thalamus, to the suprachiasmatic nuclei, and also to the superior colliculi. Information goes to the superior colliculi is for the visual reflex. Information goes to the suprachiasmatic nucleus is for the 24 day and night cycle, circadian rhythm, right? And then the third pathway goes to the thalami that ultimately will go through optic radiation and onto the primary visual cortex. So that's a summary of the pathway for visual perception. What I want to do is the physiological component, which is very important down here, the second this panel. Phototransduction of light. So what we have is on this picture. Let's focus on here. When there's no light, this is counterintuitive here. When in the dark, our photoreceptors are more active than in the presence of light. And this is how. In the absence of light, there's a molecule called rhodopsin. And that rhodopsin contains a pigment called retinol. It's sensitive to light, but in the absence of light, what's going to happen is cyclic GMP is a lot in the cell. So in this absence of light, nothing destroys the cyclic GMP. Cyclic GMP, it's one of another example of a second messenger. Lowercase c, uppercase G, uppercase M, uppercase P. So cyclic GMP is acting as a signal, a chemical signal, activating from the inside of the cell a sodium channel. So sodium channels are open, allowing sodium influx. There's potassium efflux here, you know, but there's a net incoming of positive charge would depolarize the membrane. That depolarization goes picked up by the voltage sensitive calcium channel, inducing calcium influx, releasing neurotransmitters. These neurotransmitters binds to the receptor, generating graded potential on the bipolar cell, which is here. Bipolar cell in return generates more graded potential going toward the ganglion cells, allowing the ganglion cell to go to the threshold and fire an action potential in the dark. Now, when light, the presence of light strikes the rhodopsin, activating the retinol molecule, what's going to happen is when light strikes the process, a G protein called transducin, TRANSDUCIN, this G protein gets activated. Every time light strikes rhodopsin, G protein is activated. More specifically, the alpha subunit of the G protein activates a membrane protein acting as an enzyme called phosphodiesterase. And that enzyme, phosphodiesterase, destroys the cyclic GMP. So it basically decreases the concentration of cyclic GMP in the cytosol of this photoreceptor cell. And what's going to happen without cyclic GMP, the sodium channel shuts closed. So there's no sodium influx, yet this leak channel potassium keeps going out. So by losing a net positive charge, the membrane hyperpolarizes. When the membrane hyperpolarizes, it cannot activate the voltage sensitive calcium channel, no calcium influx, less neurotransmitter being released, less graded potential being produced by the bipolar cell, less graded potential generated by ganglion cells, less action potential. So a decrease in signaling allows us to perceive light. And this is unique in vision, right? So in the dark, absence of light, we have much more activity going on in our neurons in the retina. So the retina is more active in the absence of light than in the presence of light. And yet we have adapted a way to perceive light, despite the fact that there's less depolarization involved when light is present and striking the retina of our eyes. And on that note, I'm going to end the lecture here for today, and then I will talk to you again on the next recording. Until then, take care. Be safe out there.